The fourth time is the charm. Okay, so we've been talking about this, specifically our adaptive immune response, right, as it relates to B cells and T cells, this specific response that we have. And in order for this response to happen, we have to have cells that present antigen. So this was a picture from the very first website that I sent you guys to, right? And so it's showing a cell, in this case a dendritic cell, engulfing and processing that pathogen. So they're destroying it in the process. It could be a bacteria, it could be a virus, it could be any number of things. And then it's going to present it. What they're not showing you in detail here is what they use to present, right? So if this is something they took from outside, something that doesn't get inside of our cells, so a bacteria that stays outside of our cells, right? What class of molecule do they use to present this with? MHC2, right? So remember, antigen presentation happens with MHC class 2. And another clue as to this would be MHC class 2 presentation that's happening here is that what T cell the dendritic cell is communicating with. This is a helper T cell, right? But it has the CD marker what? 4, which is the only one that can communicate with MHC class 2. In this process here, this antigen presentation by the dendritic cell, who else could present antigen other than a dendritic cell? B cells will do this in their activation process. Who else? Macrophages, right? But dendritic cells are our main presenters, right? They're our scouts. They're going to gather this information. They're going to send it back to our lymphatic cells, these T cells. So this T cell with this interaction becomes activated. What does that mean? So it's activated. So what? What does that mean? And it's going to proliferate, right? Because we want more than just one. And what's important about this interaction? What's happening here? Why is it binding with this one? Why does it get activated? Partly, right? It is recognizing this specific antigen here. Right? That's the important thing, because remember, each T cell is unique. Its T cell receptor recognizes a specific antigen. Same is true for the B cells, right? Theirs is just an antibody on their surface, but it's their B cell receptor. This is a specific interaction. So this T cell, helper T cell, gets activated, right? Which means that it recognizes this specific pathogen we're dealing with right now. And it's going to proliferate so that each one of those is the same, right? This is clonal expansion. They're all the same, and they're all going to recognize that same pathogen. So now once they've been activated, they could go and activate macrophages further that are presenting this antigen. They can activate B cells that are presenting this antigen. Not always the case, but most likely, yes, this happens, right? You're going to have antigen presentation. In order to get an adaptive immune response to activate B cells and T cells, you've got to have this antigen presentation. You've got to find B cells and T cells that recognize that antigen, right? Before they become activated, and they're going to proliferate. So we don't just have one cell, we have lots of cells. And then as she alluded to earlier, right, not only are we going to get activated T cells that are going to go out and activate B cells and macrophages further now, but we're also going to get memory cells produced, right? And so the second time we're exposed, usually it's the memory cells that are going to become activated, and that's why our response is so swift, as opposed to trying to find a naive, meaning a T cell or a B cell that's never seen antigen before. Right? We've got to find those special matches. Make sense? So right? For antigen presentation, dendritic cells, macrophages can do both. Both antigen presentation to helper T cells and antigen presentation to cytotoxic T cells known as CD8 cells. Now CD8 cells, when they recognize antigen on the surface of a cell, what are they going to do to that cell? They're going to kill it, right? They're cytotoxic. So this is the defense we have against stuff that gets inside of our cells. So how do we display, how do we give this information? We have MHC class 2, right, for stuff outside of our cells, right? And we're going to activate helper T cells that will probably activate also B cells so that our response is extracellular. We produce antibodies, right? But if we have an intercellular pathogen, be it a virus or a bacteria, we want that type of response. So the way that we get that type of response is the dendritic cell presented on MHC class 1, right? And they're showing you that here, right? They're different colors. It's really hard to see in this diagram. It's not my favorite diagram. But remember, these dendritic cells are out in our tissues. They'll travel to the lymph node or the spleen to be able to present this information. They're almost like the scouts, right? That's my nickname for them. They're out gathering information and bringing it back to home base. So anywhere, right, the closest location for B cells and T cells for it to present antigen, it's going to go to depending on where it's encountered this stuff in your body. So this drawing too from that first website that I sent you to covers this process related to B cell activation. So again, a B cell has to come in contact, and B cells, unlike T cells, don't have to have antigen presented to them in order to become activated, right? But sometimes they do. Themselves have to present the antigens they've come in contact with. And who's going to help them with this? The helper T cell, right, as the name implies. All right, so that's the step they're showing you here, but they're not showing you the detail here, right? What is actually happening is that this cell, right, has interacted with an antigen, and then it's going to bring it in and process it, right? And it's going to present it on MHC class 2, right? And that's going to communicate with this helper T cell because helper T cells have CD4, right? So that's an interaction that's happening right here that's not drawn in. This interaction, this helper T cell has already been activated. It is now going to activate, it's already been activated probably by a dendritic cell. It's going to now help activate this B cell. This is called a T-dependent antigen, an antigen where we need this step. Other antigens are T-independent, right, where they can go straight here, skip over this step, and go to the next step of proliferation and differentiation. So this particular diagram is specifically showing where you need that T help, right? It's a T-dependent antigen. Whatever the antigen may be. In this case, it's showing a bacteria. So this is important too, because remember we said that different B cells will make different antibodies, right? We have the 5 different classes of antibodies. What's the first class that they're always going to make? And especially if they don't have T cell help, they're only going to make this class. No, IgM. IgM is the first one we always make, right? The most common switch we make is to IgG, right? That's the most protective antibody, can do most of the protective outcomes. But in order to make that switch from IgM to IgG, you need T cell helper T cell help, right? To communicate to that B cell to make that switch. So remember, plasma cells are going to do what? Yeah, they produce the immunoglobulins, the antibodies, right? They actually secrete them, right? The B cell has, and this isn't the best drawing, right? Because remember, antibodies have 2 antigen binding sites, right? And they're only really showing 1 here. But on the surface of a B cell is the B cell receptor. So instead of just having it on its surface, a plasma cell increases its rough endoplasmic reticulum and actually secretes the antibodies. Right? So they go into your bloodstream, they go into your lymph, they can travel into your tissue fluids. Right? In the case of IgA, which you make associated with your mucosal membranes, right, it enters into your mucus. You also make it in your mammary glands to go into your breast milk, right, for women. You produce it in your tears, in your saliva, right? So the helper T cells in those areas will stimulate those B cells to make IgA, right? Where ones in your lymph nodes and in your spleen, mostly they're going to produce IgG. And then in some instances, right, you get the production of IgE, right? And that typically is a response to stuff that wouldn't necessarily hurt you, what we refer to as allergens, right? And so this, we refer to this as an over-response, and it's in our disorders unit that's coming up next, our next topic. So remember, again, whenever a B cell or a T cell is activated, we always get those memory cells, right? And so, again, and this is just one of the protective outcomes, once antibodies are produced, they'll bind specifically to that specific pathogen, the bacteria in this case. One of the outcomes, one of the benefits of that is it tags them for destruction. What's the term that we use when an antibody binds and therefore it tags it for destruction by phagocytic cells? Opsonization, right? So one of the protective outcomes is this opsonization. And so this guy is going to gobble them up and destroy them. The second time, right, your body is exposed to this specific bacteria, typically it's going to be those memory cells that are activated, not more naive. So that's why the response is swifter and faster. And how do we take advantage of that? What can we give somebody to give them a first exposure before they even get sick? Vaccination, right? So flu season's coming up, right? Don't forget. And I think it's today. Did you guys get an email? I always try to remind my students. The health fair that they're having, they usually have at, I think it's in the gymnasium here in building 22. I erased the email. I usually save it and resend it to you guys because usually you read emails I send you as opposed to the ones the entire campus sends out. But they do free immunizations for other immunizations you need. I think you have to pay for the flu vaccine like $10 or something like that. But they are doing it on campus. And I want to say I think it's today. So if you have time, go check it out after class. Definitely, you know, take advantage of not having to go to the pharmacy or the doctor's office, right? So, this is a diagram from your book. And this kind of summarizes diagrammically the whole thing. And so again, I have you guys working on, and if you came in late today, I made the announcement that I extended the bonus assignment where you watch the videos. Remember that you either have to hit submit or there's a text box that opens up in order to submit your assignment. It depends on what device apparently you're on, right? What way you're trying to do your assignment. So there's either a blue button that says submit or there's a text box and a blue button that you, after you type, you submit. So I extended that to next week, and I've extended the exam date to the following week. Okay. So this is a really good diagram, and as I said, and as I do in that activity, I find for immunology, it's good to draw things out. I mean, you don't have to draw perfectly. I mean, they could be rudimentary drawings like I did in that activity. But going through that process and talking through that process and drawing out that process can be very beneficial, right? So see how much you can remember and draw out on your own of these processes. So Remember, as I said, dendritic cells are these important scouts, right, that are going to collect information, and they're going to bring it back to sites where your lymphocytes are located, where these B cells and T cells are located. Remember, they originally came from your bone marrow, and for your B cells, they are educated between self and non-self there, and then they're going to travel to your secondary lymphoid organs. So what's some examples of an organ where your lymphocytes hang out? Lymph nodes, right? We have lots of those, right? So we have that protection all throughout to filter that lymph through the lymphatic vessels. What's another organ that you actually could live without? Spleen. Kidney, not the case. Definitely can't live without without at least one of them, right? Your spleen, right? That is filtering your blood, right? So it's chock full of macrophages and T cells and B cells to respond to anything that's in your blood. So you can survive without that, but then you risk, bloodborne type infections happening and not having that level of protection. Some of the other functions of your spleen do get taken over by your liver, which is why you can live without your spleen. Your appendix, your appendix is another place where we have a large concentration of B cells and T cells and macrophages. And this is another place where, it probably at one time was where we would process certain types of foods that we don't process anymore. So unfortunately, sometimes stuff will get trapped in there and it could get inflamed because of the large collection of white blood cells there. So sometimes that'll be removed, right? So it's none of those things, again, that you can live without, and actually can be a detriment to us sometimes when stuff gets stuck in there and it gets inflamed, because it could rupture, and it is just an appendage off your large intestine, right at the beginning of your small intestine to large intestine, near the cecum section. And so if that bursts, then contents of your intestinal tract are leaking into your abdominal cavity. And that's why it can be very serious and very life-threatening. Because again, remember, bad stuff can go through our digestive tract as long as it goes through, right? In one end and out the other, no problem. It's when it breaches that barrier that we run into problems, right? So your lymph nodes, your tonsils, right? Don't forget about those, right? And then all your mucus-associated lymphoid tissue, your skin-associated lymphoid tissue. These are all areas where there's lots of lymphocytes, your B cells and T cells. And this is where the antigen presentation is going to happen. But these cells, too, can leave these areas once they've been activated. So activation will happen here, proliferation will happen, but cells will leave, right? Because typically the virus-infected cells, right, that your cytotoxic T cells need to go destroy are not in your lymph node or your spleen, right? They're going to have to go out and hunt these guys down. So one of the things I do like about this drawing is notice that once activated, you've got memory T cells. These guys are going to stick around in the secondary lymphoid organs and tissues, right? The memory cells. Because that's where activation is going to happen again if we see something again, right? So dendritic cell comes along, right, and very quickly interacts with these memory cells, and we get a very quick response. So for each of our lymphocytes, our cytotoxic T cells, if they're activated, our helper T cells, notice we'll get memory cells that'll stay here, and for our B cells. Again, helper T cells are going to help out other cells. They'll help out macrophages and making them more powerful in their killing. They'll, as you see here, help out the B cells to become activated. An antigen itself can activate a B cell. But for B cells to class switch and stuff like that, right, produce a particular class of antibody, they have to have interaction with helper T cells. So remember, the active class gets another name, right? They're called plasma cells. And this is because their rough endoplasmic reticulum, right, their cytoplasm really increases significantly to be able to produce millions upon billions of these antibodies within seconds. And these two, the plasma cells will even leave this area and produce antibodies right on site until the infection clears, right? Once it clears up, all these cells, these helper T cells, these cytotoxic T cells, these plasma cells, they'll go through apoptosis, which remember is programmed cell death without what? No inflammation, right? This is normal cell death. So these guys, they've served their purpose. We don't need them anymore because who do we have in case this problem happens again? Memory cells. And you could, even for a virus, because remember, antibodies can be protective against viruses when they're outside of our cells, right? You could get all of this happening when you have a virus infection. You could get all of this happening if you have a bacterial infection, especially if it's one that gets inside of our cells. You want a cytotoxic T cell response. You want a CD8 response. Make sense? Right. So in some diseases, right, we go sometimes, depending on the person's genetics or how the disease is, we may only produce antibodies, right? Or we may only do a cytotoxic T cell response, right? But the response is going to be tailored to the pathogen and to how your body chooses to respond, right? So there's a lot of variability in this. So she said, does this happen every time? And I was like, no. Right? But it's going to happen a lot. There is the potential there for it to always happen. Does that make sense? Okay. So this is when it works, right? So just as a review, remember, I even added in the classes, but IgG, right, can do all of these things. This is our most protective antibody. It's in this characteristic Y form. It can neutralize viruses and toxins. It binds to them so they can't bind to us, right? A virus, first step, it's got to bind to us. If an antibody is coating it, it cannot bind to us and enter into our cells. Same thing with toxins. They usually have to interact directly with our cells. If antibodies are binding to them, then they cannot attach to us. IgM can do this, right? That mega big one can bind to them and stop them from being able to bind to us. IgA can do that in our secretions, right? It can help neutralize them, keep them from attaching to us. And it's main reason why, this year I'm actually going to try not getting the flu shot, but getting the flu mist up my nose, right? Because the hope is I'll produce more IgAs, right? Because it's going to be my mucosal membranes that are going to get exposed to that vaccine. So hopefully I will produce a lot of IgA, right? So in my mucus, hopefully I'll have IgAs that will trap the virus before it even gets into my body, right? Opsonization, right? Remember, it's the FC region of the antibody that our phagocytic cells have attachments for. Now, for phagocytosis, macrophages and dendritic cells, and who else is a major phagocytic cell? Neutrophils, right? But neutrophils don't do antigen presentation, right? They're just basically our killers, right? Our suicide bombers. They're the strongest killers out there. That's their main job, phagocytosis and destroy. Where our macrophages and dendritic cells go one step further. They'll do antigen presentation. They can gobble up a lot of antigen and still survive. So notice too with complement activation, right? Complement proteins can interact with antibodies, but there's only two classes of antibodies that have this ability, IgM and IgG. And again, those are our major ones. Remember that MAC complex that forms? What does MAC stand for? Membrane attack complex, right? That can form from complement proteins from the activation of this system. The name of the activation when it's activated by antibodies is referred to as classical. When C3b binds, that's the alternative pathway. And then there's one other activation pathway for complement. Lectin binding. Remember, we're going to get an inflammatory response when we get activation, and there are two complement proteins that you need to know their letter designations and numbers that are involved in the inflammatory response. C3A. C3b is the binding one, right? That one is the opsonizer. The other half is C3a, and then C5a. Those are the two, right, that you need to know that are involved in the inflammatory response. Immobilization, right? So IgM and IgA definitely in our secretions can help immobilize. Again, stop them from being able to adhere to us. Same is true for IgM, right? All of them can cross-link up, right? IgA, IgG, IgM, really good at clumping them up, right? Either causing a precipitation for very soluble molecules or agglutinating large things like bacteria. Only IgG can do this response, right? And this is the last thing that we're going to focus in on as far as our adaptive immune response is the other lymphocyte, which is the natural killer cells. And natural killer cells, unlike lymphocytes, unlike B cell and T cell lymphocytes, do not have receptors that specifically recognize a specific antigen, right? They don't have specificity. In this case, they're inducing the death of this infected cell because antibodies are binding to this cell and recognizing the pathogen that's within this cell. This process is referred to as antibody-dependent cellular cytotoxicity. This natural killer cell will not kill this cell normally, right? It has to have some type of signal that says that this cell is a bad cell. In this case, it's an antibody of the class IgG only, right? So they have receptors for that Fc portion, that stem portion of our antibodies. Binds to it and will kill that cell. The other class of antibody that we haven't talked about much yet is IgE. And IgE will attach to mast cells in our tissues, and of course the cell is holding on to it by the Fc portion. So the antigen binding portions, the arms, are sticking out, right? So especially for allergic individuals like myself, my mast cells are potentially coated in antibodies against the things I'm allergic to. So literally within seconds of inhaling pollen that I'm allergic to, these cells will degranulate and release the histamine, and we get that inflammation response, right? What we refer to as congestion as it refers to our nose. So why do we have this response, right? For some pathogens out there, this was an appropriate response, right? The unfortunate thing is that it can be a very bothersome response when it's against things that aren't going to harm us, like pollen, right? And dander and things like that. So my stuff is out of the order that I wanted to. So natural killer cells. So they have two distinct protective roles. One of them we just reviewed, right, is this antibody-dependent cellular cytotoxicity, right? You definitely need IgGs for that. And so this, unfortunately, is one of the ways, and we'll talk about in disorders, hemolytic disease of the newborn, right? It's when the mom have made anti-Rh antibodies and they are IgG. Remember, IgG can cross the placenta, right, and enter into the baby. So then they bind to the baby's red blood cells, the mom's antibodies, and the baby's natural killer cells recognize these antibodies even though they're mom's, and they'll kill the baby's own red blood cells. Hence hemolytic, destroying red blood cells. So the other type of protection is not having MHC class 1 on the surface of a cell. So remember, this is how all of our cells communicate with cytotoxic T cells. All the time your cells are presenting antigen, self-antigens on its surface. But cytotoxic T cells don't kill them when they're presenting self-antigen, right? Because they can recognize that that's us. It's when, say, they get infected by a virus and they're picking up pieces of that virus and putting it on the surface, then our body would recognize that. The cytotoxic T cells would recognize that. Some viruses, unfortunately, shut down that mechanism of antigen presentation inside of our cells. So our cells no longer present MHC class 1 on the surface when they're infected with some viruses. So we have a backup to that. It's called the natural killer cell. When a cell is not expressing MHC class 1, and they're supposed to, right, there must be something wrong with that cell. So therefore the natural killer cell will kill that cell if it's not expressing MHC class 1 on the surface. Not doing any antigen presentation at all, they're going to destroy it. So those are our two main protective outcomes of our natural killer cells. Eliminating cells that are coded in the antibodies of the IgG class. So the antibodies are recognizing that there is something specifically wrong with that cell. Or when they're not doing antigen presentation at all, they're not presenting MHC class 1 at all, the natural killer cells will eliminate them. So we already talked about diversity. So we have actually finished up adaptive, and I have not finished up the Connect assignments for adaptive yet either. But hopefully you guys have been working on and have completed innate, so that way if you have questions. I'm not going to officially start the next lecture, but I want to give you guys some pointers to look over before Friday. So I'm going to go ahead and open that one up. If I could open the right unit. Disorders. So you'll notice that there is a table in your book that summarizes some of the important aspects of the 4 different hypersensitivities that we're going to talk about. For my PowerPoint lecture, I devised a streamed down version, right, of just what you need to know as it relates to each one of these hypersensitivities that we're going to talk about next time. And you'll notice that they number them 1, 2, 3, and 4. And for testing purposes, I'll include the numbers, but I'll have the full title, right? So we're really focusing on the title, not necessarily just the number designation to the type. So we'll talk about immediate IgE mediated. And in this title, it tells you a lot about what's going on, right? Especially for that one. Immune complex. Do you guys remember what I said an immune complex is? It's antigen and antibody, right? When you have that binding, you have that cross-linking that's happening. That's what we mean by an immune complex. So, as always, you know, tables are really good for information. I may or may not play the animations that are associated with this. And remember, those links, right, are in Canvas as well. I would suggest looking at them ahead of time, right? It's a good way to familiarize yourself with the material before I lecture on it, as well as, of course, looking at the PowerPoint lecture, reading in your book, right? Preparing yourself ahead of time. Right? So we'll start the disorders lecture on Friday. And